Hi everybody, my name is Sebastian Böcker. I'm from the Friedrich Schiller University in Jena, and I'm going to talk about ZODIAC, and we don't have much time, so let's jump right into the topic. And our task is the annotation of molecular formulas of small molecules from ISMB data. We assume that we have complete LC-MS runs, and we want to do that de novo. That is, we don't want to look up the possible answer, the candidates from some, say, structure database, but we really want to consider all possible molecular formulas chemically feasible. And that makes it obviously much harder since we do not restrict ourselves to a small set of candidates. We consider them all. And you might ask why, but I would rather ask why not, because we know that we don't know a lot. In particular, we don't know a lot of structures which are out there. So why would we assume that novel molecules have the same molecular formulas as already existing molecules? That simply doesn't make much sense. So we should really do that if we want to go for novel compounds. Here's a results teaser. In blue is Sirius, our current state-of-the-art method for this. We have evaluated against several other methods. There are not too many which can really do that de novo. If you know others, please let me know. Anyways, Sirius is so much better than these other methods that it's really just worth to evaluate our new tool against Sirius. The new tool ZODIAC improves the error rate, which is given here, by pretty much 50%. It also reduces the error rate below 10 and usually below 5%. On most data sets, my tool is different. It's gut microbiome data, and the data quality is not as good because of a broad isolation window, so let's not think about that too long. But even there, ZODIAC improves the molecular formula annotation rate. Dendroids is special because the compounds are very large. I mean, large, small compounds sounds funny, but it means compounds around 700 or 800 Dalton, and we see that there in particular, the annotation rate improves dramatically, and that was actually the reason why we developed ZODIAC in the first place. ZODIAC builds on Sirius, so let's first talk about what Sirius does with tandem MS data. Sirius also analyzes isotope patterns, but that is not of interest here. If you have a tandem MS, what Sirius tries to do, or what it does, is it tries to annotate the fragments in the tandem MS. Let's assume we have a candidate molecular formula for the precursor ion. Then we try to also annotate all of these fragment peaks with molecular formulas. There are sometimes multiple possibilities. And what we do when we compute such a fragmentation tree is we decide for one of those possibilities, because it's pretty impossible that they are simultaneously. So we annotate those and we tell you what the fragment dependencies are. That is solved mathematically as a maximum a posteriori estimator. On the computational side, on the algorithmic side, it leads to an NP-hard problem, but we can compute them relatively quick, which we have to do because we have to solve this problem repeatedly for every single tandem MS. Now, we don't do that only for one molecular formula candidate. We consider all molecular formula candidates that might explain the precursor ion. Then we have a best fragmentation tree for each of these candidates. And what Sirius does, I mean, it also considers the isotope pattern, as I told you, but let's ignore that for the moment. What Sirius does, it would now choose this one because it has the highest score and tell you that this is the molecular formula of the precursor ion. But in ZODIAC, we consider that we have a complete LC-MS runs, and that means that we have probably measured many structurally highly similar metabolites, because in such a biological run, obviously enzymatic reactions transform one metabolite into another. And that's what, actually is interesting for us, but we can also use that to improve the data analysis. And if we have two structurally similar metabolites, and we assume to know the structures here, obviously they are not known to us, but let's assume we know them. What we see is in the fragmentation spectra, they share a lot of peaks, but they don't only share peaks in the spectrum fragments, they actually these are the same fragments. They have the same molecular formula. So the annotation of those peaks is also identical. In fact, the same is true the other way around for the losses, but I'm not going into the details of that. Yeah, it's pretty much clear what you have to do for that. And that means we can now compare any two candidates. And if this is the correct annotation for this spectrum, and this is the correct annotation for this spectrum, and these two are structurally very similar, then these two should share many annotations of fragments. They should be identical. Whereas if you grab any two random trees, and they are hopefully then, or let's assume they are wrong, then these two should not share that many annotated fragments. Obviously, they will share some, but not as many. So the score, if this is correct and this is correct, should be higher. And with that, we can now improve or increase the probability that this is actually the correct molecular formula annotation, if we already know that this one is the correct molecular formula annotation. And we do that for the complete network. So we have hundreds to 10,000s of spectra, and for each of them currently we use like 50 molecular formula candidates, and between any such two we now have some similarity, which is used to modify the likelihood that this is the correct annotation, or the posterior probability, I have to say. And what we use for that, because it's an NP-hard problem, we use Gibbs sampling, which has nice properties. It tells us also posterior probabilities, and we can use it to compute marginal probabilities, because that's what we really want to know. How likely is it that this is the correct molecular formula for this tandem MS? Unfortunately, Gibbs sampling is pretty slow, at least if you use it based on matrix multiplication. So we put a lot of algorithm engineering into that, and now it's really, really fast, but I can't talk about that because I don't have the time. So here are evaluation results again. We also tried something different, which is we tried anchors, which is library hits, and that didn't improve much, so I'm not going to talk about that. You already saw the results. This one is noteworthy. We also have the problem that we have to assign adducts to each tandem MS, which means we don't know if it's protonated or if it's a zodiated ion. So if we give that to the method and if we don't do any errors on that part, then the error rate of ZODIAC on this data set decreases to almost zero. Also, and that is shown here for the dendroids data set, we improve the error rate across the complete mass range. So we also found some new molecular formulas in here, which are just recorded here as a list, and not all of them will be correct in these different data sets, but we verified 2 of them manually, and we are absolutely sure that these 2 are correct. We don't know the structures, but we don't have to. We just know it's a novel molecular formula, so there's also a novel structure behind it. They contain nasty elements, bromine, chlorine, iodine, but that's just what happens if you go into, say, diatoms or something. With that, oh no, this is all available from Sirius 4.4. You can download it, and then using Sirius is just a click of a button. And with that, I come to the end. I don't have time for repeating much, but I want to tell you that Marcus did much of the method development and implementation, and Louis did the biological evaluations. Thank you.